this pattern of the neuronal cell types in the ventral spinal cord is controlled by another graded signaling system, which is shown in this diagram here. So this blue circle has SHH written on it. So the signaling system here has a strange name. It's called Sonic Hedgehog signaling. And so you might ask why a gene that controls many aspects, as you'll see, of nervous system patterning acquires such a strange name. And there's a long history to that, but the recent account is that a scientist at Harvard, Clifford Tabin, when he discovered with others this gene, decided to call it after Sonic the Hedgehog, which had video game notoriety at that stage. So what I would just say to you is that, is there a better reason for becoming interested in neuroscience and maybe even becoming a neuroscientist than the possibility of discovering a gene that encodes some important brain function and naming it after your favorite video game? So I will leave Sonic here in a reclining state just to remind you of the excitement of gene discovery in brain science. So Sonic Hedgehog controls all of these neurons, and it does it in an interesting way through this gradient. So these red cells require high concentrations of Sonic Hedgehog signaling, while the blue V0 cells require a low concentration of Sonic signaling. So this gradient acting at concentration thresholds makes the difference between these neuronal types. Remarkably, this graded patterning signaling system is true not just in the spinal cord, but throughout the central nervous system. Sonic Hedgehog controls the pattern of half the cell types in the central nervous system. So if we look in this image, we're now showing in blue on the right this stripe of Sonic Hedgehog that you saw. And if we take a section through the level of the forebrain region, which contains the neurons that are going to give rise to the retina, which control vision in the eye, you can see that the position of the eye field is furthest away from this Sonic Hedgehog gradient, whereas other cell types are much nearer to the Sonic Hedgehog gradient. So you might imagine that given this graded signaling system that defines at different concentration thresholds, twofold differences in concentration, that this signaling pathway would be extremely sensitive to perturbation. If you get the concentration wrong, then you would have a consequence for differences in cell fate. And I just want to give you, with a demonstration, if you like, a demonstration, how one might think about changing the Sonic Hedgehog signaling concentration, and then I'll talk about the consequences of that. So here we're looking at this same image where the eye field is generated in a hedgehog-dependent way, but far from the source of hedgehog here. So imagine what might happen if we start to strip away the concentration of Sonic Hedgehog for some reason or another. Then the cells that are most dependent, that need the highest concentration, would disappear first. And as you gradually decrease the sonic signaling concentration, those neurons that are generated at more ventral positions would disappear, and there would be a progressive shift in the position of cells generated at more dorsal regions. And eventually the limiting extreme of this would be that in the virtual absence of Sonic Hedgehog signaling, you would lose perhaps all but the most dorsally generated cell types, which in the forebrain is the eye field. And so this raises the question, are there conditions in which these concentration-dependent hedgehog signaling pathways really are affected, and do they have an influence on brain organization and development? And I will show you just one example that this is in fact the case from a set of animal studies, but then I'll talk briefly about human genetics that argues the same thing is true. So this slide may be a little bit shocking, but it's perhaps, I won't leave it up for long, but this is what we're going to be looking at, is an example of cattle or sheep in the wild who eat a certain plant, the corn lily or hellebore, which contains a veratrum alkaloid, a chemical which blocks hedgehog signaling through its receptor. And as a consequence, what happens is most of the ventral cell types, so the progeny of the animals, the newborn lambs or calves from parents that have eaten those plants, show this sort of defect. They show this cyclopia here. So on the right, we're looking at a single midline eye in much the same way that I showed you on this panel here. So this is an example in the animal kingdom of blockade of hedgehog signaling leading to brain patterning abnormalities. Now, one also sees this, unfortunately, in human cases where there's not ingestion of an alkaloid from a plant, but genetic lesions in the hedgehog signaling pathway. And those patients suffer from a syndrome called holoprosencephaly, which again is associated with the progressive loss of neurons from these ventral regions as a consequence of the blockade of hedgehog signaling. So in this way, what I've tried to do in the first part of this lecture is give you a sense of how one goes from a uniform population of cells, begins to diversify those cells to make the 5,000 or so cell types, neuronal types, that we see in the human brain.
Think forward. Think Research Channel.